Yeah, so we're stopping at 10:40.
Yeah. Okay, so
Don't forget to sign in if you did.
so this is going to be a lot less mathy and interesting, but I did compensate with a lot of nice, colorful images. So hopefully that'll be nice, and we'll have time. We'll get to the heavy stuff toward the end before lunch.
Wait. Start again.
I just put the mic on.
I did.
Okay.
Do I need to turn this on or something?
Is it on?
Is it red?
It's red. It's fine. So I'll start. Okay.
That joke again.
Yeah, it wasn't good enough. And now I need to think of a completely different joke. Yeah. So I'm going to talk about generally human evolution, fairly recent human evolution, and a lot of the kind of interesting things that happened in the past kind of 500,000 years in human evolution. So I think kind of since the dawn of man, we've been very much interested in where we come from. And for ages, we've only been able to kind of examine this question kind of philosophically and maybe theologically. There was no real science behind it. And only kind of in the mid-19th century, we were able to kind of pose actual scientific questions about our origins, obviously due to the theory of evolution pioneered by Charles Darwin at that time, and also by the first kind of physical findings of ancient humans. You see these skulls and skeletons that look undeniably human, but still somewhat different. So it was pretty consistent with Darwin's theory at the time that we didn't just appear from nowhere and were part of a continuum. And as time kind of passed, more and more of these findings popped up, and we kind of created this interesting thread of human evolution in the past, something like 3 million years back, kind of toward our common ancestor with chimpanzees. And while we find more and more of these findings, it's often tempting to try to kind of find their place on the path, but that's not very straightforward. First of all, the dating. I have some dates here, but they're not very accurate. But the more complicated thing that Darwin already realized, kind of an earlier version of Darwin, realized that all of the observations we make in nature should be associated with tips of a tree. They shouldn't really be put on a path, even if they're ancestral. And so all of these kind of nice-looking skulls should actually are part of these kind of branching processes somewhere in the middle of these branches, right? So even if we know the date, we don't always know if it really appears in the direct line of lineage from present-day humans back or on some branch. So for most of these, it's not really consequential. I know some people argue, even for the very deep ones, whether some of these are really kind of direct ancestors or distant branches. But as you go back in time, probably kind of the distance along this little branch versus the big distance that you have to make the humans is much small. But for the more recent findings, Neanderthals and other kind of and other human kind of forms that we find in the past, say 500,000 years, it's very, very significant. This divergence is very significant, and you have to kind of account for it. And so that's going to be the focus of my talk, is going to be in this period and how it became in the past, say, decade or even less than a decade, became much more complex than we originally thought. So we're going to zoom in kind of on this period in time. So most of the talk is going to be on Neanderthals. So here we have a nice kind of Neanderthal specimen, and you can see even just by kind of the naked eye that it's very different. If you put a Neanderthal skeleton next to a kind of modern human, it's more wide and kind of more thick-boned and has a bigger skull, something like 1.5 times, 50% larger than a modern human skull. And it also has this kind of very characteristical perturbation in the brow area. So when people started finding these skulls, this is the first Neanderthal found in the Neander Valley in Germany. At first, they were tempted to think that these weren't necessarily a different group of humans. Maybe it was just kind of diseased individuals. But as you find more and more of these, you see that they're pretty spread out in time and in space. So the kind of geographic distribution of Neanderthals goes all the way to the western Iberian Peninsula and all the way east to kind of central Siberia. So it's a pretty wide range, and you find Neanderthals dating as early as some 400,000 years back, and the latest specimens are dated around 30,000 years ago. And that's about the time where we stop finding Neanderthals, and we see only kind of modern human specimens. So they died out for some reason that we still don't quite understand. So and during all of this time, we see, at least like the early part of this time, we don't see any kind of overlapping modern humans in the same area. So that gave rise to the hypothesis that Neanderthals are not our direct ancestors, but are a sister group, sister species. Now, obviously, not clear how much of a distinct species they were, but distinct group of humans that evolved somewhere in Europe around 300,000 years ago, and our ancestry is kind of parallel, and mostly, at least until something like 70,000 years ago, was focused in sub-Saharan Africa, with the little stretches here, maybe in the Mideast and North Africa. And I had to actually update this slide. This used to say 200,000 years. Now it says 300,000 years because of the latest finding in Morocco. So you see, I couldn't update this. This is kind of hard copied in, so it was difficult to read. So you see sometimes 200,000, sometimes 300,000. These things are very fluid. They're going to change as we make new discoveries, obviously. But that's about the time. So for a long time, for almost 500,000 years, these two branches were pretty separate. We don't see a lot of overlapping kind of physical evidence of overlap. But then around 65,000 years ago, humans, our ancestors, modern humans, started expanding in Europe and in Asia, and kind of entering Neanderthal territory. And so there was a period of something like 30,000 years where there was at least potential territorial overlap between these two groups that have been diverged for something like 500,000 years. So it's pretty kind of, in our perspective, it's a pretty kind of unique period of time. And ever since kind of this picture was kind of set in people's minds, there was this question of whether this kind of potential for interaction between these groups, whether it actually materialized, whether there was cultural interaction and physical interaction, and obviously kind of sexual interaction between these two groups, and whether there is any consequence to us, to human populations today. So that is one of the big questions about Neanderthals. Probably the other one, which is even bigger, is what made them kind of disappear. How come that they disappeared? Is it related to human expansion? So we're making some strides in understanding this a bit. So interbreeding between kind of these archaic forms, Neanderthals and modern humans, has been, as I said, a very basic question in the study of Neanderthals. But the physical evidence is very, very difficult to find, because you have to realize, in order to find some physical evidence of interaction, you have to have a site where you have Neanderthal bones and human bones that you can actually date to the same, pretty much the same generation, like a joint burial site or something like that. That's very rare to find. The only thing that kind of comes close are some of these findings that they see in various sites, actually in Romania, are these skulls that kind of have hybrid features. They have some Neanderthal features, some modern features, and they've been hypothesized to be maybe some evidence of again, sexual interactions between Neanderthals and modern humans at that area. But again, it's very controversial because it depends on your interpretation. There are not a lot of features here. There's not a lot of data, in a way, to make really statistically significant inference about physical interaction just from like one or two skulls. On the other hand, genetic data, if we can actually get it to work right, is going to be much better for this, right? Even with one or two individuals, there are going to be a lot more data in genome-wide sequence data, and it's direct. We can have actually direct evidence of interbreeding from genetic data because we can actually find the consequence of interbreeding. It's not going to be circumstantial. So there was a lot of drive in for at least, I think, 2 decades to get this to work, with one of the kind of flagship questions being whether there were genetic interactions between our ancestors and these extinct human groups. So, yeah.
This is just a curiosity. With the genetic data that we have, is it possible to say if it tended to be, human females and Neanderthal males, or the other way around?
Yeah, maybe we can discuss it later, because I'm going to talk about the signals. Okay. Maybe that question will. Right. Well, there is no clear answer to that, but it's not impossible, I think, to be able to say that, but it's not going to be easy. Dan's talk last week mentioned a lot of the kind of complications of being able to sequence ancient DNA, and how we, computationally, we need to take into account some of these complications. So I don't want to get too much into it. It's not really relevant to what I'm going to talk about today, because I'm going to assume that we already have high quality data, which has not been the case for, it's really only in the past maybe 5 years that we have really high quality ancient DNA data. But the main issues are, at least for this type of study, contamination is probably the biggest issue, because when we sequence a bone, we sequence many things, the actual DNA of the person who had that bone in them, say 100,000 years ago, but also a lot of bacteria and maybe other humans or animals that were in contact with that bone. So it could be scientists, but it could even be just humans who encountered that bone maybe 1,000 years ago and touched it, right? So we have to be very, very careful in making sure that we get what's called like endogenous DNA. And so we're, again, getting better and better at that, and there are methods of doing that. I'm not gonna talk too much about caveats there, but because, again, a lot of them have actually been solved in a lot of these high-quality sequences that we get. Other issues are these errors in sequencing we get because the Cs get deaminated, and then some of them get sequenced as Ts. Again, we can address it computationally or chemically, and there is the issue of fragmentation. The fragments of DNA get shorter with time, so again, it kind of it becomes a problem if we want to sequence kind of very long haplotypes. But all of these issues are pretty much addressed. And I want to just acknowledge kind of, what I think are really the two people who pushed this field very, very far. Svante Paabo, obviously from the Max Planck Institute in Leipzig, who really drove a lot of the developments in the kind of earlier phase, and Mattias Meier, who's a group leader, again, also in Leipzig. He's really in charge of a lot of the advances that have been able to, that made it possible for us to sequence high quality DNA from a lot of the individuals a lot of the studies that I'm going to mention now. So the first time that we actually got a glimpse of like what we can say is a Neanderthal genome was 2010. when the first draft Neanderthal genome was published, and it wasn't a genome in the sense that we think of today, because it wasn't a specific individual. It was actually DNA taken from 3 different bones, so probably 3 different individuals, and sequenced at very low coverage. So it had to be pooled to gain some coverage of the entire genome, but it was genome-wide. Like, a lot of the genome was covered by at least, say, 1 or 2 reads. So you could get a reasonable read of the entire genome. And obviously, this was examined for various things. And one of the main questions was to see whether there is evidence of, again, interbreeding between Neanderthals and mostly, again, hypothesized humans outside of Africa. So how can you address these types of questions? And that's going to bring me back to some of the things that I talked about in my tutorial talk. In order to make a lot of these inferences from genomes, especially from small numbers of genomes, and here we have a single genome and several modern human genomes, in order to make useful inferences on population history, we're going to have to think about these genealogies in the middle. So just to remind you, what do I mean by these genealogies? It's actually this ancestral recombination graph, which is this perturbing tree along the genome. So remember, every site in the genome is associated with a tree going back in time. That's kind of the evolutionary tree of that site. And the trees change with ancestral recombination events. There are these slight changes as we go. So this tree is kind of perturbed as we go along. That's going to be kind of the process that we're going to think of. So in my tutorial talk, I talked a lot about demography inference and this method I developed, GFOX, for doing this, inferring demographic parameters from these unlinked alignments along small numbers of genomes. So we're going to also see some analysis of that, and I'm going to mention 2 other methods that are kind of genealogically oriented. The D statistic, or the ABA-BABA test, which I'm going to describe just in a few minutes, which is a very common test for admixture. So it's not typically described as a genealogical test, but I think it's a very useful way of thinking about this test. And ArgWeaver, which is a more sophisticated machinery to do some actual sampling of these args and more, again, sophisticated inference about ancestral events in this ancestral recombination graph. So what is the D statistic, or the ABA-BABA test? It's very, very simple, and the main objective of this test is to look at a small number of samples. So you have your kind of potential source sample. It's not necessarily literally the source, but it's kind of the hypothesized source of gene flow. Here, Neanderthals. There is an outgroup, chimpanzee. It has to be an outgroup for all the populations we consider. And we have 2 possible in-groups, genomes from different populations. So it can be, say, present-day Africans or present-day Eurasians, right? A single genome from each of these, and we look at an alignment of these genomes. We think of them as haploid genomes. So if it's diploid, we just take 1 allele. So we have 4 alleles in each site, and we look for specific sites that have these patterns. So think of it, you have this chimpanzee allele, and you're looking for sites where only one of the other 3 genomes shares that allele, and the other 2 genomes have an alternative allele. So you have it's either the Neanderthals share this allele, or the African in this site, or the Eurasian. So you have 3 types of sites that we type as BBAA. A is going to be the chimpanzee allele, so it's always going to be A. So you have BBAA, ABBA, or BABA. And you can associate each of these sites with a hypothetical genealogy at that position. So it doesn't necessarily have to be the genealogy, right? So because it's a biallelic site, so we can think of a single mutation occurring somewhere ancestrally, and it sits on a certain branch, and that kind of splits our four samples into 2 pairs, right? So we're trying to see which which pair is the derived pair, right? Has the derived allele. So here in these sites we have the two present-day humans with the derived allele. It can be the present-day European. Here it's the present-day European and the Neanderthal share the derived allele, and here it's the present-day African and the Neanderthal share the derived allele. So we have no confidence that this is actually the genealogy here. There can be double mutations or everything, but the idea is that you can think of these sites as associated with these genealogies with some noise. So all this test does is actually count these two types of sites. And why is that? So this type of site, the BBAA, is going to be the most common because, again, we expect most of the trees to have this shape, where two modern samples are going to be more closely related. And these two types, we still expect to see them at some portion, just because of incomplete lineage sorting, just because these two lineages don't have to coalesce during this time, and if they don't coalesce, then they're free to coalesce with the Neanderthal branch at this ancestral population, right? And if that happens, then we can have a shared derived allele with Neanderthal and Eurasian, or with Neanderthal and the African. So you have these two sites, and you actually see them quite a lot in these genomes. And the question that this test asks is whether we see one of these sites in excess compared to the other. And the nice thing about this is that under all reasonable models of error, noise, demographic histories that kind of control incomplete lineage sorting, unless you actually have some direct interaction between unless you actually have violation of this tree structure, I'll say that formally, you don't expect to see a significant excess of ABBA versus BABA sites. And if you see a significant excess, then you say, I kind of reject the null hypothesis of a tree like this, and there has to be some other interaction. And so what they saw when they looked at this draft Neanderthal genome is indeed, yes?
But what you're saying implicitly assumes that the sort of early population was a mixtic in some sense, right? That there was no population structure.
Yeah. So if there was, so you can explain these things with population structure, but then you have to kind of, it's not just population structure, but you kind of have to assume that you mean population structure here in the early, like in the ancestral modern human branch. Yeah, you're going to have to kind of assume that that it dates all the way back here, and again that there is some affinity. So all you're saying is that kind of the ancestral European population has more affinity to Neanderthals than the African. It can be, so it doesn't necessarily have to be a very recent branch here, but in a way there's some point of interaction that ancestral Africans didn't have. So, so yeah, I'm gonna show evidence that you can kind of use to say that this is a recent event, that it happened after divergence, really after divergence. But the ABBA-BABA test doesn't do that, right? So it's just a very simple rejection of a simple null kind of tree-like structure. Again, people tend to overinterpret it sometimes as real evidence, positive evidence of introgression. I'm going to show that it's not that simple. That sometimes there are alternative explanations. Okay. So this is pretty much this is pretty much the main test. There were additional tests done in that paper to establish kind of this gene flow from Neanderthals to Eurasians, but this was the basic one, and the ones that's the most popular today when people look at other species and try to examine to see whether there is gene flow or introgression. And it's very, very powerful for a few reasons. First, it's simple. People like simple tests, and it kind of, you can think of it as you have your null, which is the tree, and you have kind of two alternatives, right? There is gene flow from your source to one target, or gene flow from your source to the other target, depending on which the ABBA or the BABA sites has an excess, right? So you have kind of three hypotheses that you're trying to kind of choose from. So it's very, very simple. it doesn't take a lot of data, and you can use it with low coverage data, very noisy data. All of that kind of goes into the basic statistical framework where a lot of this noise is going to be uniform. It's going to cancel out with the ABBA and the BABA. It won't give you excess of one versus the other. And it doesn't make any assumptions about the ancestral demography, which is very important in the early stages because you didn't know anything about demography of Neanderthals, and you knew little about demography of modern humans. We know now much more. So it was very, very useful, and that's why it's very widely used. And using this basic test and additional, again, evidence in this early work with the draft genome, we were already, I mean, not we, but others were already able to establish a pretty strong evidence of this, again, gene flow from Neanderthals to modern humans at a rate that affects something like 1 to 3% of the genomes, of the genome of pretty much everyone who has a non-African ancestry somewhere in their history. So it has a pretty wide, small, but very wide effect on all non-African human populations. So that was the draft genome, and then the next step was to try to get a real genome, a real high coverage Neanderthal genome. And again, the scientists at the Max Planck Institute were very lucky to find in this very, very small bone found in the Denisova cave enough DNA to sequence at 30x coverage to get a really high coverage genome. And the interesting kind of development was that it wasn't really Neanderthal. So when you compared it to the draft Neanderthal genome, you saw that it was quite diverged. So it was, if you draw like a population tree, it belonged to a population that diverged from Neanderthals something like 300,000 to 400,000 years ago. So you couldn't really say that it was Neanderthal, and we don't know anything about, this is pretty much all we know. There's also a molar that we have. You have like few bones of these individuals from this Denisova cave, and even today we don't know much about this population. We know that they're significantly diverged from Neanderthals, so we just call them Denisovans. So we don't know if they were really just located in that region or spread around. Yeah.
In the 3 bones of the previous analysis,
yeah.
Do you know if they were male or female?
Yeah, I think they know. I don't know specifically here. I think they know. I think you know. You see, I don't know if they knew before sequencing because there are a lot of bones, and I don't know if they could associate them, with something that's characteristically male or female. Yeah, I think you get reads from the X chromosome, but again, it's so low coverage that I'm not entirely sure that they really know. This one is a female because you have high coverage and you can easily see. Yeah.
How did you determine the numbers, the 400, the 300? How did you determine by mutation?
With mutation. So I'm going to show like our models that do this, but here they were simple models. You look at the difference, you estimate the ancestral size, population size, and you can get an estimate of the time of divergence of the population. It's not just average distance, genomic distance. It factors the ancestral population size, but this is a fairly crude estimate. It's not a real. It wasn't a very well-formed demographic estimate, but it holds today even as well.
Now, all the analysis before just ignored recombination. It has nothing to do with recombination, correct?
Well, the ABBA-BABA test kind of assumes that different parts of your genome have different trees. So it uses recombination, but doesn't have to model it because it assumes they're independent and it's separate sites. There is no model of recombination. Yeah, there's no model here yet. I'm going to talk about more model-based.
It ignored recombination here, no?
The ABBA-BABA test?
Yeah.
It doesn't ignore it because if you have no recombination, there is a single tree. You have to have recombination so that the tree will actually change, but it doesn't model recombination in an explicit way. Okay. So the nice thing about this genome, first of all, it's a high coverage genome. Even if it's not Neanderthal, it's closely related enough to Neanderthal so that we can reaffirm again this finding with a high coverage genome, as scientists did. So you can use it still to find evidence of this branch. So you don't have the source is never really the population ancestral to the one that we're looking for. It's always something related to it. Even here, the source is probably, branches somewhere here. So the fact that this branches 400,000 years and not maybe 200,000 years is kind of it's a numbers issue. It's not a really significant difference. So you could still find this evidence, and interestingly, you can find evidence of interbreeding specific to ancestors of Denisovans or populations very closely related to these Denisovans in present-day populations in Papua New Guinea and also some in Southeast Asia. So it appears to be like a separate event that contributed kind of gene flow from these populations to, again, the ancestors of modern humans that live in this region of the globe. Yeah?
When you say more closely, do you mean more closely than Neanderthals or more closely than other
Yeah,
populations?
no,
that
so
aren't
for this
common?
type of thing, you do like an Abba-Baba test with Denisovan, say Papuan, African, and chimpanzee. You don't use Neanderthal, but when you do it with Neanderthal, you don't see, so you see kind of an excess relative to what you see with Neanderthals.
Those are more close to this than Neanderthal.
Yeah. So for instance, when you do it with Neanderthal, you don't, and you look at a variety of present-day humans, you don't see a lot of variation. But here, when you do it with Denisovans, you see all certain a peak in these populations. You see much more kind of a stronger signal in these populations than the rest, which again was a signal that it's a separate event. Again, additionally, all of these things have to be, again, examined by more careful methods that I'm going to discuss later to actually, understand more about these events. And only 2 years later, in 2014, another bone found in the same Denisova cave was sequenced at high coverage, and this time it was an actual Neanderthal that's called the Altai Neanderthal because Denisovan was taken by an entire population now. And again, by sequencing this, first of all, again, people find the same things. It's always good to find the same things with additional, data. We have whole genomes, but still, if the genomes are somehow warped in some way or contaminated in some way, it still can affect the results. People are very careful so that it doesn't. But any additional genome that captures the same signals is very important when you're talking about really small sample sizes. And additional events that we're gonna, so again, you see kind of this very detailed map that we're gonna return to later that really starts to get dense. A lot of these events of interaction between kind of our ancestors and these archaic humans. But before I dive deep into that, I want to talk about something else that you can do with these high coverage genomes that relates to understanding more about these events. So what you can do, this is a very, very simple kind of attempt at this, and there are better models to do this, but you can kind of map the alleles that in each genome that seem to be shared like these ABBA sites compared to your Neanderthal in different genomes. So for instance, if you do this with the present-day African, you see very few of these, obviously because you take the African as an outgroup, so you're expecting not to see many. And when you do it with a French, this is a French genome and this is a Han Chinese genome, you see many more. And here, if you squint, you can kind of see there's slightly more in Chinese than in French, and you can kind of argue that maybe there's a stronger event or an additional event in East Asians, and I'm not going to get into that. But the interesting thing, it really starts to get interesting once you do this with genomes, with modern genomes that are ancient. So these are genomes that belong, these are bones that belong to early modern humans. This is a femur from Ustishim. It's dated about 45,000 years ago in central Siberia. And this is a mandible bone from the Oas cave. That's that cave in Romania where they found these hybrid bones. So both of these, the Ustishim, we actually have a full genome and we can do this genome analysis. This bone, scientists were only able to get like I think, what is it, 2 million kind of bases, SNPs, pretty much along the genome. So you could do this mapping with these genomes, and you see with this Ustishim individual, you see significantly more sites, and they're much more clustered. And this clustering means that, again, you were closer to the point of admixture. These sites, these are actually haplotypes that are, as time goes, right, in the first generation, we expect half of a chromosome to be Neanderthal and the other half to be human. And as time goes, these Neanderthal haplotypes will get broken up. So you see, as we get kind of closer to the point of introgression, we do expect these blocks to be bigger, and they do get indeed bigger. So this Ustishim individual is pretty close to the point of, to at least one point of actual interbreeding, and this Awas individual is really very close. This is an extreme example, so that's why I'm showing chromosome 12 here. Pretty much half of the chromosome is covered by what seems to be a Neanderthal haplotype. And again, by trying to model the length of these blocks, you can get an estimate of something like distance of something like 4 to 6 generations between this individual and a Neanderthal individual. So it's very striking evidence that at least this individual really is the closest that we have to actual evidence of interbreeding in action. I don't think we expect to see much better than this. So again, using the distribution of the sites that are shared, you can get much better evidence of the timing. And if you use these ancient modern human bones to get DNA, you can get closer and closer to that event and get better estimates of the date.
So cluster, what do you mean by cluster?
So you see, this is not a model, right? This is like hand wavy figure thing. But here you see, so these are all sites where this genome is closer to Neanderthal than to any, say, other modern human. And you see that they're very, very tightly clustered. If you run an HMM on this, right, you'll get that this is probably a block. So there are better models of doing this, but so this is just kind of a visual representation.
This is just positions.
But you don't see it exactly. But if you look, but in most places of the genomes look like this. They're very, very sparse. This is a very clear clustering of these shared alleles with Neanderthals. Yeah.
You don't know the values.
We'll talk about this offline. But you can actually model this and use this to actually infer the time of shared ancestry, and you'll get a very, very recent time if these are your observations. Yeah. Yeah, as I don't know if it's specifically the same skull, but it's in the same cave. So this is a mandible. I don't know if they have a full skull for this one, or it's like. Yeah, so I don't know if it's a single. It's probably always more complicated, but at least it's very, very close. If you assume a single kind of Neanderthal ancestry, then it's this. If it's more, then maybe it's. But still, yeah. I don't know if the modeling took into consideration multiple ancestries. So. Yeah.
So, chunk of, as the chunks get diluted away as time goes by. So they're a good indication also for the timing of the event. But is there anything else besides here you go and then you're diluted away over time that would make these sites concentrate in non-uniform ways around?
So there's obviously I'm not going to get into that, issues of selection, right? So some places are gonna the introgression is not going to be accepted as well as others due to natural selection, and you do see that. So you do see that with more long range, that say next to genes you see a depletion, and in some places you see actually evidence of positive selection. There are these. We discussed it in Journal Club. You should have come to Journal Club. We're still continuing. It's not too late. Huh?
Can you tell how much of the drop from 6 to 9% to 1 to 3% is driven by selection versus?
Yeah, so it's difficult. So now we have more ancient genomes, and you actually see some gradient. It's mostly actually driven by these two genomes. So there is not really a continuum of ancient genomes, but you pretty much see the percentages go down, and the percentages you don't expect to go down unless you assume either natural selection, so you can explain it. You can look at the gradient and try to explain it by strength of selection, or again, there could be a diluting effect if the population that received the gene flow was small, but then kind of mixed with like a larger population coming from, I don't know, maybe further in Asia. But it doesn't seem to be likely, because it seemed like there was kind of a burst of gene flow that affected all of like the ancestors of all present-day Eurasians. So yeah, so it's difficult because there are several confounding factors, but there does seem to be an effective selection in reducing the actual fraction. But here, I'm not concentrating on the fraction, but the clustering. The clustering should only be affected by recombination and less so by gene flow. Okay, so I just wanted to, it's an interesting point. It's going to be related more to something I'm going to discuss later, but it's, again, not just, it's going beyond ABBA statistics and looking more at how these sites cluster in haplotypes. And that's going to be important later on. We're going to talk about inferring even more complex events. So this is pretty much the picture, since we have these two high coverage genomes, the Denisovan and Neanderthal, and we have, again, low coverage sample belonging to this kind of European Neanderthal branch. So there are a lot of arrows here, and I want to make kind of some order. So most of them, you see all of these arrows going from either Neanderthal to Denisovan to this big expanding human blob. So you can think of them, they're all unique and interesting, but they're kind of an artifact of the same thing, of human expanding out of Africa and expanding their territories east in Asia, west in Europe, encountering these populations and mixing with them. So it's different instantiations of the same thing, but it's kind of the same story. There's obviously a somewhat different story here. So you do see some gene flow between, again, the ancestors of this Altai Neanderthal and the ancestors of the Denisovan, which seems to make sense if they were pretty much in the same region. But there's this signal was very, very kind of curious. You can even ask yourself, what exactly does this mean? So what this means is that there is evidence of gene flow from a population that seems to be much more diverged than all of these. It's more than 1 million years ago in the tree, that gave some gene flow. There is some low-grade gene flow into Denisovans from these populations. So you can ask yourself, where do you actually get evidence of this, right? We don't have the sample here at all. So how can we even be confident enough in this signal? So again, you could do this with a pretty simple ABBA statistic, and this was the first evidence of this event. So how does this go? You do an ABBA test with Denisovan and Neanderthals as your in groups and a present-day African as your out group. And when you do that, so you expect to see, again, more BBAA sites, but you don't expect to see any difference here. But the observation is actually that you see more ABBA sites than BABA sites. So you'd be tempted to say that there is, the kind of obvious explanation, that's not the obvious, but the standard explanation in this scenario is that there is some interaction between Africans and Neanderthals. It seemed, I think, so I don't know exactly the history of this finding, but what I kind of extrapolate is that it seemed unlikely that there would be such interactions because the ancestors of Africans live all in Africa. The ancestors of Neanderthals, at least in this branch, were in Europe. There was no really point of interaction between these two populations. So another alternative explanation is this explanation. So you could get, if you have a population that's split up here that gave some gene flow into Denisovans, it would also lead to an excess of these types of trees, right? Because with this type of tree, you still get Neanderthals grouping with Africans and Denisovans as kind of the outgroup. So even a low grade of kind of this gene flow would give the slight excess that you actually see in genomes of these ABA sites versus BABA. So you don't even have to have a sample here. But again, so in a way, it just emphasizes this deficiency of the ABA-BABA statistics, that all it tells you is that this tree, you can kind of reject this simple tree, but then you never really know if there is interaction down here or something complicated up here. So what they did in the paper is actually ran some simulations with both kind of scenarios, and they were able to pretty convincingly show that the simulated data fit better when they simulated this super archaic gene flow than when you simulate something like this, an interaction here. So that was their conclusion, that that you have this event and that it's not some interaction between some gene flow from Neanderthals to Africans or from Africans to Neanderthals, which seems unlikely. So that's pretty much where we come in. So we were interested, even before really closely examining it, was before that paper came out, to do a really full model-based demography inference on this data and to see what we can find. So our starting point was, again, to use GFOX on the data, which is very well suited because there were small numbers of genomes, and it's kind of very, it can accommodate pretty complex models with a lot of gene flow to infer population sizes, split times, as well as rates of migration. So we did that with the two published genomes, and we also had access to high coverage sequence data from two additional Neanderthals, but only restricted to chromosome 21, because it was a capture technology. It wasn't a whole genome shotgun, so it was a capture assay from chromosome 21 that covers about half of that chromosome. So it's only 20 megabase of sequence. It's not a lot of sequence, but it was enough to get maybe some weak signal from these kind of more European Neanderthals. The reason to use this was there was a risk that this Altai Neanderthal is kind of at the edge of the range of Neanderthals and might not be like a bona fide Neanderthal. We wanted to make sure we can find the same signals in these more European Neanderthals. We also at that time had a lot of present-day human genomes that we could use to kind of understand the different interactions. So the first thing we looked at is, so we ran GFOX on that data, and we could, again, get unified demographic model for Neanderthals together with modern humans. So here you have it focused on Neanderthals with the split times and population sizes. So the first time you can actually get these split times with a model-based approach. So this is an approach that assumes, again, you assume a constant population size in each of these branches, but you accommodate for these population sizes and the changes as well as gene flow when you estimate these divergence times. We get something that's very similar to the kind of cruder estimates obtained before, but the confidence intervals are smaller. I don't know what to say about that. I mean, it's possible that we're overly confident about our estimates, but I think the other side of that is that using kind of an explicit model, you can know your uncertainties better than when you use a more ad hoc estimator. But there's not something very new or surprising here. When you look at effective population sizes, so again, it's not very surprising, but you see, so when you just look at these genomes, you see a very low rate of heterozygosity. So you know that we're talking about smaller effective population sizes, but it's unclear whether there was a certain bottleneck in a certain time and maybe possible expansion, or whether it was really long-term effective population size. And when you look at it in this model, you see that there seems to be like a gradual reduction in population size. So this ancestral population has something like, say, 7,000 individuals in it, and then you have a drop, like a factor of 2 drop after the split into Denisovans and Neanderthals, and it drops even further down here. Again, we start getting very sparse data on these two branches, but you do see, if you kind of again squint very hard, you can see maybe some signs of slight expansion on the Vindia branch. Yeah.
When you write the number of individuals, you mean like the number of actual humans that live during this period?
It's a number that captures a lot of things. So it's numbers of individual and mating structure. It's an effective population size. So that's the scale is number of individuals, but it's number of individuals if you assume a random mating population. Yeah, so it's not really number of individual, but in a way it's comparable. So you want to think of it as an estimator of your variability maintaining engine in that population somehow.
So GFox does model that.
Yeah,
factor.
yeah, but it models it as one parameter. It doesn't really have per population. It's a one parameter representing the size. Yeah. Exactly, and you see that it really contrasts with the large population sizes that you see in present-day humans, especially in Africans. So this is really interesting, and it's very characteristic to these archaic humans, that they lived in these small population sizes. Again, not just during like their decline, but throughout 500,000 years of their existence as a separate branch. So it's interesting, at least. But it really gets interesting. What we were really interested in is gene flow, to see, first of all, whether we can capture all the previous signals, because you always expect that, but you never really know because it's a different method. It has less power than the D statistic, which is very, very targeted. Everything can kind of go wrong, but it went pretty well. So here we had to do. It's not as clean as you see here. We actually had to do a separate analysis for each of these populations, but we can then put like all the migration rates in this big matrix, and you see the things that light up are pretty much the things that you expect, especially down here. So you see these two rates are from Denisovan to the Papuan and the Han Chinese, so that's, again, these branches that were previously observed, or various elevated rates from these European Neanderthal branches into the ancestors of present-day Eurasians. And you see again this signs of gene flow from the ancestors of Neanderthals into Denisovan. So all of these things were previously known, and it's nice to capture. And actually the numbers also pretty much coincide with the rates that were assumed at the time. But the one thing that didn't quite fit is this gene flow from these African genomes into the Altai Neanderthal. So first when we saw that, we said, there was that finding of super archaic gene flow in Denisovans, and it was actually a result that was when we found that it was actually a result of this Abba-Baba type test of kind of increased shared derived alleles between the Altai and the Africans. So it's the same type of thing. We might be capturing the exact same type of thing. So we actually modeled this super archaic gene flow. You could do this pretty easily with GFox. You just add another branch to your population tree. It doesn't have to have a sample here. And you model gene flow, and you actually capture this low-grade, it's small but significant low-grade gene flow from this ancestral branch into Denisovans, but it doesn't make this thing go away. So it seems like it's not enough to explain this. Now at that point we were still not sure, because when you use these sophisticated models, there are all sorts of reasons why the model might prefer this explanation versus this, and it might not be a real thing. So we wanted to make sure before we actually say that there is another point of contact kind of to add to this tree, that we actually see something real. And to do that, you actually have to look at the haplotype structures I'll show you next. So before that, I want to kind of show you the difference. So there is a difference between, so the basic observation again is that you see more Abba sites in this test than Baba. So there is more sharedness between the Altai Neanderthals and modern day, present day humans in Africa than with Denisovans and present day humans in Africa. And the question is whether it comes from kind of this ancestral branch or whether it comes from a more recent sharedness between Neanderthals and the Altai. And you could, if you could actually kind of date these two these two coalescent events in this tree, you could try to distinguish between these two, right? So super archaic introgression will lead to kind of pushing back of this node. It will be up here instead of being more here where you expect it. And modern introgression would take, again, this coalescent and pull it down here. So we wanted to see whether we see some evidence of ages of these, again, events or shared haplotypes. But it's very difficult to get ages of coalescent events in this kind of in these gene trees, especially if you're thinking about like these unlinked trees that we were sampling with GFOX. So with GFOX, we were actually sampling these trees. We could take the ages that we were sampling and use them. But that would be pretty much unusable because there really is not enough information just for mutation alone to tell you about the ages of coalescent events in the tree. To get that, you really need to use something about haplotype structure. You want to say, even here, again, without a model, we see that the fact that there is a long stretch of sharedness here is an indication that the coalescent time had to be recent. If it was deep, then it would be broken by recombination. Because it's recent, it's not broken by recombination, and we're able to see a long stretch of it. And to put it in more model-based term, we will really need to get kind of the ancestral recombination graph. So we want to know how these trees change along the genome, and hopefully we will be able to show evidence. Here you can see evidence of a long shared ancestry between two of these individuals that's not broken because you don't get recombination events. To break it, you have to have these two recombination events that actually fall on the branches, on the short branches of kind of that ancestry. So the length of these shared haplotypes is the signal that we're looking for. And to get that, we actually have to kind of model the ARG in some way. And the only way we knew how at that time was to use ARGweaver. So ARGweaver was, I don't have the citation here, it was developed by Matt Rasmussen, postdoc in Adam Siepel's lab, where I was a postdoc as well. And he developed this really neat and sophisticated method that, so you can't really infer an ARG because there's no way, there's no confidence, you can't tell with high confidence what the ARG is for a given set of genomes, but you can sample reasonable ARGs kind of from the posterior using this method. So it's an MCMC, Markov chain Monte Carlo sampling method that kind of efficiently and effectively samples ARG from the posterior. And yeah.
Could you try to do like more simple, not parametric thing by looking at serial correlation in the Anablava test?
So I think there are things that are done. I think Sriram is here. No, he does things like that. But we wanted again. This was I can't justify. This is a very heavy gun. I don't know if we can fully justify using this heavy gun and not a simpler gun. But you have to, as you see, the signal is pretty subtle. I'm not sure you'd get. It's not as clear cut as this. When you have something as clear cut as this, you can probably use something a bit less sophisticated to find your differences. When it gets more subtle, I think you have to have more modeling in it. But I hope to convince you that the modeling is not confounding this, that it's not a modeling artifact. So the idea is, again, so the demographic model is very simple here. That's something, you have to make compromises. It's a very sophisticated method. So you make two compromises. One is that you take this whole coalescence with recombination model and use a Markovian approximation. So the model is not actually here. It looks as if it's Markovian, that each tree depends only on the previous tree. With the full model is not really like that. There are long-range dependencies, but it works pretty well. The Markovian approximation approximates most features of the ARG pretty well. And another probably more problematic simplifying issue is that we model the ARG kind of in a single population. We don't assume any structure. So that's obviously a problem because these are very diverged samples. And we didn't know that we're going to get a signal, but we got a signal. And in a way, it kind of, if we had, I think, modeled the full demography, we would have gotten a stronger signal here. So it's somewhat approximate, but it was the best thing that we could do at the time. So the idea is, again, you get these ARGs, you get samples of these ARGs, and then you try to use them to estimate ages of these events. I have some time. Okay. So what are the ages that we're looking at? We are interested in each arc that we're sampling.
How long do you think you're talking?
What?
How long do you think you're talking?
As long as it goes, no? There is lunch afterwards. I'm not going to hold anyone here, but now I have. I was waiting for somebody to stop me, but isn't that how it goes? Okay, I'm wrapping up. I thought it would go unnoticed because of all the nice, colorful pictures. Wasn't able to distract you that much, huh? Okay, so I'll get to the kind of punch. So the idea is to, so again, the main idea is to look at these trees where your Altai branch kind of joins the tree of present-day Africans, and the Denisovan branch joins somewhere, coalesces somewhere ancestrally. It can coalesce in the tree higher up, or it can be kind of an outgroup. And you want to measure this age, and then you want to do kind of the same when you flip Denisovan and the Altai, and you want to compare these two and see if you see differences. And when you do that, so mostly you see kind of the same times. If you look at deeper times, you see most of them are pretty consistent, but when you look at the really kind of more recent coalescences, then you see, again, at these times that are kind of more recent than 200,000 years, you see a clear excess of the Altai Neanderthal coalescences that you just don't see with Denisovans. And that was our signal. That was the thing that we were looking for, and we didn't know that we would actually find. So the numbers are something, because of all the modeling assumptions, you don't want to put too much weight on the numbers. But again, this is something, and we, again, use simulations as well to convince ourselves and others that you can't get this excess under other kind of possible explanations. So this really, what this means is that with the Altai Neanderthal history, you see more of this, you see kind of a slight excess, but a significant excess of these very recent coalescences that you just don't see with Denisovans. This is something you don't expect under the alternative hypothesis. But we were also able to kind of look at it the other way around and see that there still is some evidence of this super archaic introgression. When we look at the ages of when Denisovan or the Altai are kind of act as an outgroup. And you see when you look at that, then the Denisovan ages seem to be a bit kind of more pulled to the older ages than the Altai. But this is a very, very subtle signal. So in a way, you can say that we find evidence of both these events, this super archaic introgression in Denisovan and this introgression of modern humans into the Altai. So this is our main contribution, this additional branch, and it's somewhat more complicated because, as I said, there is no direct point of interaction between these populations. So it's probably a population that is somewhat related to modern humans, but was able to migrate out of Africa and maybe lived in the Middle East, maybe in nice place like this in Israel. This is a site where you actually find modern-looking humans around 100,000 years ago. And you also find Neanderthal findings there, but they're not at all from the same age. So it's a very wide, you have a very wide timeline. So you don't find any physical evidence here, but it's a site that's attracted humans for 400,000 years. So it's likely sites like this are kind of candidates of where they might have met more than 100,000 years ago. So just to recap, I've shown all of these kind of genealogically oriented population genetic methods, and just to kind of again reiterate this issue that genealogical thinking is very useful in population genetics. And again, in terms of these mixture events, it's going to get more and more complicated with more samples. And in a way, it's not really a tree. So while these populations lived in pretty much isolation in our minds, there were enough interactions that they actually acted as a single species, and there was continuous breeding at low grades between kind of these two separate populations. And we see this evidence the more we look at the genome sequences. I'd like to thank my collaborators and funding, and thank you for sticking it, for half an hour more than you had to. Questions over lunch.